as AJ has just said, Emily and I are going to be presenting a webinar about the protein sequences and functional annotations that are stored in the UniProt KnowledgeBase. And what I will do in this guide, I hope, is give you an introduction to how to search for proteins, how to get the most from your searches, what kind of data is associated with entries for those proteins, and then things like the sequences and the features for those proteins, such as amino acid modifications, post-translational modifications, and other related sequence features. And then say a little bit about the tools which can be used to analyze the protein sequences, like BLAST and multiple sequence alignments. Very briefly say a little bit about proteomes, introduce the concept and how you can find them in UniProt. And then lastly, say a little about mutations and diseases and annotations of those in UniProt. So firstly, what is UniProt? It's a very large-scale resource which holds information in a freely accessible form of protein sequences and information about those, functional information about those sequences. So this includes the primary sequences and isoforms where they exist for any particular set of proteins derived from the same gene, physiological, so normal protein function, and information such as pathways that proteins are part of, reactions. So, for example, if a protein is an enzyme, its interactions, and where relevant, any role in disease. Structural information, which you've already heard quite a bit about, it seems, from the previous webinars, or you may have heard about, such as 3D structures and the topology of proteins in the cell. And as I mentioned before, talk about the analysis tools which are part of UniProt. So the website, our UniProt website, has a number of core aspects or principles to it. One can download data in multiple forms. It presents multiple different visualizations of the data in UniProt. So, for example, you can look at 3D structures within the UniProt's entry for a particular protein. You can search for proteins. There's simple searches, more advanced searches, which I'll talk about later on. And the results of a search can then be filtered and categorized in different ways. And once you've got a protein of interest from the website, there are then multiple ways to customize the view you see on screen, and also how it can be downloaded. Each protein entry has multiple different aspects to it, features, where the data's come from, its provenance, which is very important. We put a lot of effort into that now. And also groups of proteins grouped together by species as a proteome, and groups of similar proteins where they're clustered together. Where does all our data come from in UniProt? Well, the majority is from directly from the International Sequence Consortium, but we also pull in data from Ensemble and PDB. And then once that data is in the knowledge base, we present it in two main ways. We have proteins which are can fall into two groups. They're classed as unreviewed or reviewed. And the unreviewed entries represent direct sequence submissions and data directly from the sequence repositories. And these have been put through a computational pipeline. So they're annotated with automated annotations created by those software pipelines. And this doesn't group together isoforms. So you'll have multiple isoforms which are displayed together. Sorry, they aren't displayed together. They're accessible within the unreviewed section, but they're not grouped by gene. And this is in contrast with the reviewed section of the knowledge base, which, as you can see, is somewhat smaller. The number of entries in the reviewed section is over 251 million now. The reviewed entries have been reviewed by expert curators. So the curators at UniProt have pulled data from the literature and associated this with the protein sequences where there is functional data. And then they also group together isoforms which are derived from the same gene. This is a view of the homepage, and I've highlighted some of the tools that are found in UniProt, the current release that we're on. So UniProt is on the 4th release in 2023, and the resources which underlie the database. Zooming in on the search bar, this allows you to type any text, any free text, to perform a search with. It could be a gene name, it could be any number of different forms of text, a unique identifier. And then highlighted here are also links to the advanced search and a way of obtaining, if you're working with a list of accessions or gene IDs, how you can directly enter those into the website and search with those. So in this example, I've taken a very small part of a pathway. And let's say that you are interested in autophagy. These are proteins which are involved in the process of cellular recycling of materials or autophagy. And the different components of this are shown here. So let's pretend that we have gene names. We're interested in finding out more about these particular proteins encoded by these genes. There are multiple ways you can deal with this. You could just type them straight into the search bar, or you could do an advanced search. So we could type them in here and obtain a list of results. Or we could make use of the ID mapping tool, which is designed specifically to cope with a list. And in this example, I've picked 3 of the gene names from that previous slide and typed them into the dialog box, and then decided, well, what I want to do is say these are gene names in this highlighted in purple here, and I want to get back accession numbers. And also I want to restrict this by taxonomy. So I choose just human. And then this illustrates the results. What you would see initially, you'd see the result of this search. And one of the powerful things about the website now is that you can keep the results of searches. You can store them for up to 7 days. And if you wish, you can save them and keep them for longer than that. So if I had done such a search, I'd get a list of results. An alternative way of doing it would be to do an advanced search. And here you've got a much more powerful set of Boolean relationships you can use to relate different aspects of information stored in UniProt. And you can add multiple, using the add field, you can add multiple different criteria to your search to restrict or expand. You can then take these away again by using the remove button. So let's imagine that I've done such an advanced search, and I've decided just to take a single protein name, ATG7, which I've highlighted here, and I put a wildcard here and change the category, the filter of other information I'm interested in to the active site. So I want to know which proteins with the gene name of ATG7 have an active site in them. And then this is the kind of thing that one would see as the result. A list of entries which all have the same gene name. Some are uppercase, some are lowercase, but they're all ATG7, and they have active sites in them. So very rapidly, I've just run through what you can do with the advanced search and a basic search in the website. Now, changing tack completely, let's say that one is interested in a specific set of peptides that one's got from an experiment. This is a different kind of search entirely. So you've got a peptide here, which I've typed into the dialog box, and I want to know if I restrict the taxonomy to human over here, and then hit go, this would be the kind of result I'd get. So obviously this is the kind of data that you might have from a proteomics experiment. And what we've highlighted here is that there are actually 4 entries which match this peptide. 2 of them are reviewed and 2 of them are unreviewed. And they're all in human, as we've restricted it to human only. Moving on to look at the content of specific entries. If I'd say that I'm interested in the class of proteins related to the NF-kappa B pathway, so this is proteins which all have IKK. They're actually the result of a very simple text search. They have IKK in their entry somewhere. So let's say we're interested in this particular protein that's highlighted at the top here. So this is, as I said, part of the NF-kappa B pathway. It's an inhibitor of NF-kappa B. It's one of the subunits, and it's also known colloquially and has the gene name CHUK, C-H-U-K here. What I want to bring to your attention here is on the left-hand side. This shows for this particular entry, for this protein, a series of links which take you to different sections of the entry. So what the function of the protein is, its subcellular location, and so on, down the left-hand side. Another thing I want to highlight, bring to your attention here, is for this specific entry, as well as the view that you can see here, which has the underlining here in this series of tabs from left to right. Other ways of viewing the same information. So there's something called the variant viewer, which allows you to see protein sequence variants for that entry. The feature viewer, which I'm going to say a little bit about in a minute, and also the publications that underpin this curated entry, and a very large number of external links, which I'll also mention a little bit about later. So this is an example of a publication which supports the functional data that's in this entry. And there are 2 views. You can see it with the abstract hidden, or as in this case, with the abstract visible. And one little important bit of information I want to bring to your attention is highlighted in green here. What this means is this particular paper, identification and characterization of an I-kappa B kinase, has been cited in only one UniProt entry. So in this case, it's only been mentioned in this particular entry, but many papers are cited in multiple entries. And the little yellow icon here means that the entry which it's been mapped to, so this publication has been mapped to this entry, it's been mapped manually. It's a reviewed entry that this paper's been mapped to. And moving down the information on the page for IKKA, it turns out that this is an enzyme, and we have curated here the enzyme commission number for this protein. We can see the reaction participants that the enzyme uses in its reaction. and so on. It's also a protein for which has been curated its location within the cell. And it just happens to be a protein that shuttles backwards and forwards from the cytoplasm into the nucleus. And moving back to something I mentioned earlier, cross-references. These are a very powerful part of the UniProt KB database. Each entry has a very large number of potential cross-references to other external databases that have additional data. Now, moving on to ways of exploring the sequences in an entry. This is now a different entry that I'm switching to, but down the left-hand side here, for this entry, I've shown the toolbar, the sidebar, and highlighted sequence. So this is the default form that the sequence takes, but you can change this if you wish, and you can alter it so that it displays in FASTA or other formats, and then you can download it. It just so happens that this particular protein, this entry, is for the spike glycoprotein of a virus, which unfortunately we've come to become quite familiar with recently. This is the coronavirus, of course, that has been so much in our thoughts in the recent years. And in this example, what I've done is I've switched to what's called the feature viewer. So, as I mentioned earlier, this is an alternative way of viewing the data for a specific entry. And in this next view, I've highlighted this yellow rectangle here, which is then circled in blue. This shows I've selected a specific region of the protein, this little block here. And it just so happens that this is a specific domain within the protein. And because there's a structure for this protein, highlighted in green here, in this 3D flat representation of a 3D structure, it shows this specific domain highlighted. If we decide we'd like to follow this up in some way, perhaps we want to blast this to find similar proteins, every entry, there's a link within it for running a blast. And there are multiple options for blast. You can change the settings. The defaults are most commonly used, but it is possible to change the settings within the blast tool. And in this an example, I've got the results actually of a completely different blast search. The default is a maximum of 250 results. I think this might be adjustable. I'm not absolutely sure. And down the left-hand side here, I've highlighted a number of filters which can be applied to the results of a blast search. And you can see already that this particular blast search has recovered 3 reviewed entries and 237 which are unreviewed tremble entries. But there are other ways of filtering out the results. You can alter the settings here for identity. So if you're only interested in the strongest, most similar proteins, you can alter this. And likewise, e-value and score values. And within the blast, having found a match, and you're interested in the alignment of those, the protein with your query, the hit protein with your query, you can select a specific alignment, and this will pop up in the window below. And there are 2 different ways of viewing this. There's this one is the default setting, which is called the overview, and there's also a wrapped version of this, which shows you the whole thing wrapped around the screen. Having got the results of a blast, and you've got a series of hits, maybe you want to look at this in more detail. One way of doing this is to run a multiple sequence alignment. And so what one can do is select in these checkboxes the results, the hits from your BLAST search, and then run a multiple sequence alignment of those. And you can either include your query or not. It's adjustable. It's an option that one can take. And this is the window that one sees when one is setting up a multiple sequence alignment. And then this is the kind of thing that one would see as a result. And in this example, we've got a series of proteins which are all related to a human gene called parkin, or rather a protein called parkin. And one of the powerful things about the BLAST display now in UniProt is that one can, where the annotations exist, one can choose an annotation from one of the hits. And in this case, I've decided to look at the fact that the parkin ortholog from fruit fly, Drosophila melanogaster, which is what this protein is here, it has an active site annotated. So if I now highlight this on the website and select this annotation, I'd see something like this on the right in more detail in green. And it just so happens that the active site, the most important residue, is this cysteine here. And we can see that it's conserved between fruit fly, human, mouse, and rat. And this is the query sequence which was used. Excuse me. And I'm just going to take a sip of water. Excuse me. So I mentioned proteomes back at the beginning of the presentation. And this is one of the ways in which the proteins in UniProt are gathered together, grouped together. They're grouped by species. And so to go into a little bit more detail about that, I'm sure many of you will know about this, but UniProt has stored in its database over 160,000 proteomes, and it covers the complete tree of life. One thing that's specific about this is that we only have proteomes displayed or included in our database for species which have completely sequenced genomes. And each of these proteomes, sets of proteins, has a unique identifier. And this is another way of grouping together the proteins that are encoded by that genome. Moving on to the final example, how can one use the UniProt database and tools to look at disease and mutagenesis data? Well, on the home page, underneath these icons, you'd find supporting data. And one of the things that's mentioned is disease. And one can search for specific diseases, or one can look for specific proteins associated or involved in those diseases. And in this example, we've chosen Parkinson's disease, and the variants some of the variants which are known to be associated with this disease. So down the left-hand side of this view of the database, there's a section called disease and variants. And this is just one of the variants, one of the Parkinson's-like diseases which are stored in the UniProt database. For non-human species, we have a different way of dealing with abnormal phenotypes. So in many model organisms, there are very good phenotypic data for many of which might be considered not only model organisms, but models for human diseases. And so to distinguish them, we refer to them as phenotypes or disruption phenotypes to distinguish from disease in humans. And many, but not all of these phenotypes are the result of studies done with null mutants, so knockouts essentially. But we do capture data for knockdowns as well. So technologies like RNAi and morpholino data are included in the disruption phenotype segment of an entry for a protein. One other view one can have of the data related to mutations and mutagenesis in UniProt is one can see which specific amino acids are involved in a variant where it perhaps only affects a few, and in this case, it's just a single amino acid. And many model organism databases will name mutants, so we capture that information as well. Finally, I'd like to just summarize what I've given you, and that is a kind of whistle-stop overview of the information that's stored in the UniProt knowledge base, how you can perform a search, what kind of functional data and sequence information you can retrieve from the database, how you can analyze those sequences. I've very briefly given you an introduction to proteomes, and finally said something about disease and mutagenesis data, which UniProt stores. The database releases new updates every 8 weeks. They're all freely available, as I said right back at the beginning. And there are multiple ways of downloading the data. Some people do this and process the data in their own way. People, for example, who have more bioinformatics skills. One thing I'd like to say is that we always appreciate anything that users can tell us about ways to improve the website. So when you have feedback, please let us know. If you have problems with using the website, you can contact us. There's a link which will take you through to an email dialog box, and you can leave a message. You can also find lots of help in the Help Center, which when you scroll down the homepage of the UniProt, you'll find that. And also it's shown at the top of every page. There's a help link. And on many of the pages down the right-hand side, as well as the feedback section, there's a link which will takes you to help. And often this is context specific, so it'll know that you've been looking at a particular protein. And there's lots of other help information on other training and guides, which are stored in the Help Center on the website. And of course, the training section at EBI have more help and documentation on other resources. And I'm going to just mention a couple of those that are upcoming that participants might be interested in. There's one which Emily Bowler Barnett is also helping with. That's on programmatic access to UniProt using Python. I imagine there will be people in the audience who might find that interesting. And some of our colleagues are also from Interpro are talking about protein families and domains in a week's time, on the 8th of November. And I hope that will be of some use to you. Finally, before I stop, there's a huge number of people involved in creating the resource of UniProt, the consortiums based in two continents, in Switzerland at SIB, in the US at the PIR, in Washington and Delaware, and of course here at the EBI at Hingston in Cambridge. Too many people for me to mention. It's also not possible without sources of funding. Our major funding comes from the NIH, but we also have money from EMBL, from the European Molecular Biology Laboratory, from Open Targets, and from the Swiss bioinformatics resources from the Swiss government. So please direct any questions to the Q&A.
Okay. Thank you. Thanks, Paul, for today's webinar, and thanks everyone for joining in and asking your questions. Emily has already answered many questions in the chat, and I'll share these questions and answers by email as well with you by tomorrow. So now the Q&A floor is open. So Emily, if you could read out the questions. And yeah, this slide is just showing up the further webinars which are coming up in molecular building blocks of life. So you can browse through these webinars and register for those which you find interesting. All right, over to you, Emily.
Thank you, AJ. And in addition to these webinars, since AJ's not put mine up there because mine's not part of the sequence, Paul mentioned that I'm part of a UniProt webinar on the 16th of November with one of our developers, and I address access to UniProt via the API, and you don't need to know how to program. And also my colleague shows you how to do the same examples with Python. So if you want an introduction to programmatic access, please come along. So we've had some really good questions. Me and AJ have been busy typing and answering them. I've picked a couple for Paul, so maybe we'll start with an easy one. Paul, Abdul would like to know how to get a faster format protein sequence from the website. So maybe you do a live demo of just downloading a sequence.
Yeah, I'll have a go. Yes, so hopefully that is a list of blast hits you can see now. So this is a live view of the, I've done a blast and now I've got some sequences here. So now if I click on the link for that particular sequence and I want to download the sequence, there are various options. So what I did then was this link here says download on the this entry. So I click download, and then there's a drop-down menu, and there are various options. So I can get a representation of the whole entry or various specialized formats. And then if I wanted FASTA, or if I wanted that, I can either have just the displayed sequence, which is referred to as the canonical, or I can have the FASTA format for the main displayed sequence, the canonical one, and also any isoforms. So if I now click, say, canonical and isoform, and then download, on the screen, you should now be able to see a whole bunch of, actually, there's quite a few isoforms there, aren't there? So that's how to do it.
Lovely, thank you, Paul. Sorry, I've been busy typing more answers. So we've done that one. An anonymous user says, can UniProt be used to find out whether a genomic mutation, like a single nucleotide variant, affects a functional domain of the protein? For example, an active site of an enzyme.
Yes, I guess if you were to take your sequence with a variant in, and then you could do that in a number of ways. Perhaps put a blast in, blast it, and then once you've got an alignment, as long as there is a match in the database, you could then look at your variants, or alternatively, if you know already what protein you're looking at, and you know which protein should have functional information in it, you should be able to then look in the database directly and say, well, if I know what the protein or the gene's called, and obviously you know which species it comes from, there might be a curated entry which has the information in that you need. Of course, it depends on that variant actually being in that entry. So it may or may not be.
So the other thing is the feature viewer. I don't know if you want to show
Yes,
that.
the feature viewer, yeah. So if I was to go to the feature viewer, and I know what variant if I knew which specific amino acid, it might be easiest done by a blast, I suspect. But if you knew which amino acid you were interested in, you could find it. So, for example, here we've got the different colors indicate the predicted outcome or consequence of a particular variant. So the ones that are likely to be pathogenic or known to be pathogenic are this dark, not sure what color that is, plum. To me, it looks like plum. And one can filter them as well by just clicking on the left-hand side, the panel, and you can then see likely pathogenic only, or likely pathogenic or pathogenic. Does that answer the question, do you think?
I think so, yeah. While we've got you sharing screen, do you want to show people where they can see protein abundance, so expression data and a protein entry?
Expression data, yes, I didn't mention that at all, did I?
No, so there's so much data to show, it's hard to do it in one webinar, but the actual question was protein abundance is data from proteomes. Now, we currently don't import protein abundance data from proteomes. It's not a link we currently have in the website, but we do import from Expression Atlas. So if you click on the expression section, we have our own tissue specificity expression.
Yeah.
There'll be free text answer, and then you have links to organism-specific databases. So you'll have HPA for human and other gene expression databases. So get clicking, but not specifically for proteomes or proteomic data yet.
No.
Watch this space, maybe.
I mean, that is something that you're involved in, isn't it?
It is, yeah. So my background's in proteomics. We brought some PTM data in for rice, but we haven't specifically got any expression data in yet. It will be lovely. Fingers crossed. So I've got another question here. Can I blast a consensus sequence with X's in it, for example, ACXVI, where X is a whatever amino acid? So
yes,
I didn't know the answer to this, so I actually put this in Slack, and I got my colleagues, and yes, you can.
Yes, you can.
So you just put your protein sequence in there, and you put the X's where you want to put unknown amino acid, and hit blast, and it does do it.
As long as you're using the recognized, is it UPAC, IUPAC, the recognized amino acid codes, it should work.
Yep. So I learned something new today.
Yep.
Sam wants to know, when would you recommend we use InterPro over UniProt and vice versa?
It depends on your question, is my question. I guess if you're interested in a broad swathe of related proteins which share a domain of some kind, perhaps you'd go to InterPro first. And then, I mean, they're both linked heavily together. So you can jump backwards and forwards from one to the other. I mean, so for example, if we look at family and domains here, let's see, we've got a ProSite. This would be an automated imported annotation saying that this domain is ubiquitin-like. That would have ultimately come from InterPro. And then the other ones which have been manually assigned, all from publications. So you could start here, or you could go to InterPro and come the other way.
Lovely. Do we contain data about post-translational modifications of mRNA?
mRNA?
Yeah, unfortunately we don't.
No, I think if you want that, I don't know if they actually hold it, but is it RNA Central?
Yeah.
They may hold data like that. I don't know if they do, but that's one place you could try.
I mean, RNA Central mainly holds RNA data which are specifically for non-coding RNAs. But I'm sorry, others.
I wouldn't know about mRNAs then. No. Okay.
So can you blast a query sequence against a particular organism?
Yes, you can. Yeah.
Yes, you can.
If you know the taxonomy ID, that's one way of doing it. That's the most specific way of doing it. You can start typing a species name into the box when you're doing a blast, but that sometimes can be slightly tedious. So, for example, if you type bacteria, it comes up with a whole lot of matches. So it would be it's much faster if you know a taxonomy ID for your species of interest.
Right. So we've had a couple of questions about reviewed and unreviewed proteins, Paul. Maybe we should just go over this, because when we work for UniProt, we understand a lot, because we have to touch an unreviewed entry to bring it into the reviewed side of the database.
Yes.
So from a user's perspective, it can be quite confusing. So maybe we should show them a Tremble entry and then show them a SwissProt entry. Okay. I have a good example of this. If you type into the advanced search gene name STUB, STUB1.
Okay. STUB1. Just into gene name?
Yeah, just into gene name.
Okay.
So I'm actually working on this at the moment. This is how I know it's an example. So STUB1, also known as CHIP. It's a ubiquitin ligase. Yeah, I've been complaining about this all week. Paul knows. So the human and the mouse are in SwissProt, and the rat, which is what I'm working on, is currently in Tremble. Okay, so if you want to show them a rat entry.
So that's what you can see in rat at the moment. Yeah. So it's got a name, a protein name.
Yeah.
But then there are several, as we can see, just by using that gene name and looking at rat, we've got 5 different entries here. Some which might well be isoforms, I would guess, from the fact they've got isoform in their name.
So D62 is the one that matches the human canonical. So if we do a direct comparison between this and the human, so this is what a Tremble entry looks like. You have the grey icons next to the accession ID, and next to its status it says UniProtKB unreviewed Tremble entry. Yeah. And it's not got a function comment. So
yes, there's no function. As you can see, it's blank.
So, Paul, if you want to open the human while I talk, so you'll see when you look at a Tremble entry, there won't be a function comment unless it's been imported by a rule, or the evidence tags on here will be in grey because they'll be automatically annotated, which means the information is brought in by similarity or by family-driven rules, which are created manually by curators. So the biological data behind the rules is correct and double-checked, but it doesn't mean we've looked at the actual entry itself and checked it. When we bring a Tremble entry from Tremble into SwissProt, so we review it, a curator takes it out, they look at all these different sequences, they make sure the canonical is annotated correctly, and any isoforms that need annotating and bringing in are merged with the canonical. And then we also add papers and data manually, and then it's populated with more cross-references as well. So if we look at the human entry, we've got the gold icons here that mean it's reviewed, and then it's SwissProt. And then if Paul scrolls down, you can see all the manual information that's put in there with all the PubMed IDs.
Lovingly added by Emily and colleagues.
Yes. Yes, lovingly. So the entire page. So this will have everything that the Tremble entry had in it, which will have been double checked by a curator, plus manual annotation. So one of the questions was, is a good protein reviewed or unreviewed? All unreviewed proteins have biological data that have been added by sensible, logical rules. But a reviewed protein entry will inevitably have more data if there's more data out there to put in. It depends what you're using it for. Human and mouse and other model organisms have obviously got more reviewed entries than other obscure proteins and species. So it doesn't mean a Tremble entry is bad. It just means you're going to find more data that's been reviewed and manually added to a SwissProt or reviewed entry.
That sounds great. Thank you.
If that makes sense, I think. And now I'm wet. So someone wants a demo for Blast. Have you got a blast that you've run today that you can show?
I have, yes. Let me just have a look.
And while you're bringing that up, someone else has asked, we mentioned annotations by my team, but how else do we get data into UniProt? So we bring data in from well over 180 different cross-reference databases. And if you look on the homepage, the main homepage, Paul won't show you now, but if you go on the homepage, right down the bottom, it gives you an entire list. There's a link that says cross-reference databases. And there's a whole list of databases that are brought in at every release. So every single protein in the database, if there's been an update and a cross-reference database between, say, now and the last release, it'll be brought in at that release. So that's how we get the other data into UniProt. We mainly bring in from ENA and other sequence databases, and then we build the entries on that every time. So that's how we do that. And Paul has his blast example now, which I'll let him talk over.
Yeah. So this is one I did earlier. I feel like I'm a Blue Peter presenter. I'm afraid that's a very cultural specific and age specific non-joke. So this is the output of the blast. So if I was to actually go back and redo it. So this is the page you see when you want to set up a blast. So I've taken, I could have pasted in this sequence that you can see in the box here, or I could have added, in fact, I might do it now. If I take the accession number and type it in at the top here, that's one of the magic, the wonders of it is if I now, I'll just go like this, delete that, and paste the accession number in, it automatically converts an accession, which I've just pasted into the box here. It then automatically shows you the FASTA format sequence in the lower box, and you can get up to 5 sequences in here. And one other thing that we didn't cover is that if you wanted, you could load these from a text file. You can take a plain text file with up to 5 sequences in it and put those into the website from this link. So having done that, I could choose which database specifically I wanted to look at. So I could restrict it to what we call the reference proteomes and SwissProt. So the SwissProt section of the database is everything that's manually curated, everything that's been reviewed. The reference proteomes include all proteins for a sequenced genome. So that's quite a good subset of the total data set in UniProt. Or you could choose UniProt, which would be absolutely everything. And then there are various, as you can see, other options. So if I choose this and then say, well, I'm only interested in human, I can choose the taxonomy here. And of course, whilst we're trying to do this live, it's taking a very long time to decide, well, what's human mean? There we are. So now I have to actually, from this drop-down menu, select Homo sapiens, and you can see that's appeared here. There's a little link that says Homo sapiens, and this is the taxonomy ID for humans. And then click Blast, Run. And then it will then go back to the tool page and show you, well, yes, you've set up a blast search. This is its name. At the moment, it's just got this anonymous link. This link actually is very useful because you can use this to go back to this, and it will be kept for 7 days anyway. And as you can see, it's running at the moment. It tells me what the target database was and that I've restricted my search to only human. As you can see, it's still running. So we could just go back to the one I did earlier. And there are the 250 hits, which are the default. So that gives you a flavor of how to run a blast search. And you can see now it's giving me a little reminder, this little thing that's popped up on my screen. It says that the blast search I set up a few moments ago is now finished. And by restricting it to human, you can see it's only found 141 hits. If I click on that, it takes me to those.
Lovely. Thank you, Paul. Right, maybe one last one. I'm a little bit aware of time. So I've had a question about where can I see reported or predicted interaction data for a particular protein? So I'll let you pick your protein and go to the section, Paul. We don't show predicted interactions.
No.
All our information that's brought in is imported from experimental databases. The closest you're going to get to predicted interaction is probably something where the evidence is in mouse, and we've copied that comment over to human because of the orthology between the species. So if Paul goes to interaction, the interaction section, you'll see that we've got a free text section on subunit and all the data from papers. And then below that, we also have binary interaction data from imported databases as well.
Yes, so this is, as Emily just said, where it says binary interactions here, this is where there's multiple sources of interaction, evidence of interaction. I think the minimum number that will allow it to be included in a UniProt entry is 2. I believe that's right. There has to be at least 2 different sources of data which say this pair of proteins interacts. In this case, this is Parkin, and it interacts with FBX07, this particular isoform.
Yeah. And you can also search the type of interaction there as well.
Yeah.
Which can be useful depending on kind of what your question is.
Oh, you mean this drop-down menu?
Yeah, yeah.
Ah, yes, yeah. So one of the options there is binary. So these would be in the same species. I think xeno means, are there interactions which have been found with a partner from a non-human species in this case? So the other species, usually they'd be mammals, but not always. Is that right, Emily?
I think so, yeah.
Yeah, I think that's what xeno means. I have to say, I've never clicked on that drop-down menu before.
I think there's only a few proteins that have that, because obviously it depends on the database.
Yes.
Yeah. I think that's the case. Yeah.
So in fact, if I go to this one, it's a septin. It's taking a while to load. What species is it in? I think it might be mouse. Mouse or rat is my prediction.
Mouse.
Yes, it's mouse. It's a mouse protein. So mouse septin, or septin 5 rather, interacts with human Parkin. Yeah,
so that'll be an imported. So this is interesting. So this will be data from an imported source.
Yeah.
Especially since it's in that table, because if we had an experiment that we were curating from a paper and they used a human protein construct that interacted with a mouse protein construct, we would add that information to both human and mouse for both proteins. So that's how we as curators will curate that into UniProt, but obviously imported databases will make that distinction between mouse septin interacts with human, Parkin or whatever.
Parkin, yeah.
Yeah. So when you're using these pages and looking at where the evidence comes from, it's important to remember that, some data, it will distinguish between species, and then other data, because it's imported from other databases, they do something slightly different, hence xeno interactions.